Stage 1A phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase. In this, a phosphate group is transferred from ATP to glucose forming glucose 6-phosphate. Stage 2 glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme phosphoglucomutase. Stage 3 the other ATP molecule transfers a phosphate group to fructose 6-phosphate and converts it into fructose 1,6-bisphosphate by the action of the enzyme phosphofructokinase. Stage 4 the enzyme aldolase converts fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. Step 5 triose phosphate isomerase converts dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. Step 6 this step undergoes two reactions. The enzyme glyceraldehyde 3-phosphate dehydrogenase transfers one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH plus H+. Glyceraldehyde 3-phosphate dehydrogenase adds a phosphate to the oxidized glyceraldehyde phosphate to form 1,3-bisphosphoglycerate. Step 7 phosphate is transferred from 1,3-bisphosphoglycerate to ADP to form ATP with the help of phosphoglycerokinase. Thus, two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. Step 8 the phosphate of both the phosphoglycerate molecules is relocated from the third to the second carbon to yield two molecules of 2-phosphoglycerate by the enzyme phosphoglyceromutase. Step 9 the enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate. Step 10 a phosphate from phosphoenol pyruvate is transferred to ADP to form pyruvate and ATP by the action of pyruvate kinase. Two molecules of pyruvate and ATP are obtained as the end products.